I thank the APA for the opportunity to present my work on the characterization of circulating pancreatic stellate cells in pancreatic cancer by single-cell transcriptomic analysis. We have previously shown that in pancreatic cancer, not only do cancer cells metastasize, pancreatic stellate cells do so as well. Our previous work used immunocytochemistry and a small range of markers to identify these novel circulating cells. The aim of this study is to identify and characterize these cells by single-cell RNA sequencing. We used an orthotopic mouse model, which involved injection of luciferase-tagged human cancer cells, ASPC1, and cancer-associated human pancreatic stellate cells into the pancreas of athymic mice. After 8 weeks, blood metastases, primary tumor samples were collected, enriched, and processed. Matching cultured cancer and pancreatic stellate cells were also processed. To identify circulating cells, one first had to distinguish human from mouse cells, which narrows down the identities to one of the two implanted cell types. Mouse and human cells can be confidently identified by relative transcript counts and by differences in gene expression, as demonstrated by these plots. Using this general strategy, 3 bioinformatic approaches were used to identify circulating pancreatic stellate cells. The first 2 approaches rely on integration of data across samples, then classification of cells using UMAP-based methods. The third approach avoids assumptions of data integration by directly using data from cultured cell sample to classify human cells in the mouse blood sample. The Venn diagram shows that circulating stellate cells identified by these 3 approaches demonstrated high levels of agreement. This heat map demonstrates that circulating pancreatic stellate cells, CPSCs, express different genes from circulating tumor cells, CTCs. CPSCs express genes for extracellular matrix and myofibroblast proteins, whereas CTCs express genes for epithelial proteins. Importantly, none of the CPSCs expressed luciferase, the transgene marker of the implanted cancer cells. Trajectory analysis attempts to find genes relevant to the fate of cells. Pancreatic stellate cells demonstrate a bifurcated trajectory, which starts off here, where most of the cells are from the primary tumor, and then splits into 2 limbs dominated by circulating pancreatic stellate cells and metastatic stellate cells, respectively. A possible interpretation of this trajectory is that as PSCs metastasize, many remain in blood as CPSCs, although some extravasate into metastatic sites. This explains the bifurcated nature of the trajectory. This heat map summarizes gene expression differences between these 2 cell fates. Genes for collagen-1 alpha-1 and mitochondrial pyruvate carrier 2 were associated with metastatic pancreatic stellate cells, whereas genes for insulin-like growth factor binding protein 1, hyaluronan-mediated motility receptor, and claudin-7, and others were associated with circulating stellate cell fate. These genes form potential targets for future studies. In conclusion, this is the first study to identify and characterize circulating pancreatic stellate cells using single-cell RNA sequencing. Characterization of potential pathways relevant for PSC fates provides targets for future studies to elucidate the role of circulating pancreatic stellate cells in pancreatic cancer metastasis. I'd like to thank the members of the Pancreatic Research Group and the CTC Group. I'd also like to acknowledge sources of scholarship support and project funding.